Today's video we're going to take a look at the structure of DNA and see specifically what it's made of. And this is IB section 3.3 and 7.1. Now DNA is made of 4 different types of nitrogen bases. DNA is made of a couple different parts and we're going to look at those today. But what distinguishes or makes each of these different parts, which we call nucleotides, different is what type of nitrogen base that they have. And so a DNA nucleotide has different types of base, and that's what makes them different or unique. The first group is something called pyrimidines. And pyrimidines have a single ring here. These both have nitrogen in them. They're slightly different format, different shape. But these are the pyrimidines, thymine and cytosine. The second group is adenine and guanine. And adenine and guanine are the purines, and you can see here they are a double ring. And so that's what's distinguishing these two. These are both, all 4 of these are nitrogen bases, and so we have 2 different types, purines, pyrimidines. Pyrimidines only have a single ring, the purines have 2. Adenine and guanine are the purines, thymine and cytosine are the pyrimidines. Now the structure of DNA is composed of 3 different parts. We have our nitrogen base, which we just talked about, those 4 different types. We have our sugar, which in DNA is deoxyribose, and we have our phosphate group. These 3 parts make up the nucleotide. These are what each portion of that DNA is made of, is these 3 parts: the phosphate, the sugar, and the nitrogen base. And so now we can take a look and see how these are put together to make our DNA structure, which eventually becomes the double helix. If we put multiple nucleotides together, we see that they form kind of these chains. And there's a specific direction or orientation that we'll look at that describes how these are put together. Here is our sugar, here's our phosphate group, and here is our nitrogen base. The phosphate group and the sugar are connected by something called a phosphodiester bond. That's what's linking the sugar and the phosphate together. And so when we're putting multiple nucleotides together in a chain, we've got our sugar and our phosphate, a sugar and a phosphate. And so that cycle continues all the way along for the DNA chain. You can see that there are some numbers here, this 3' and 5'. DNA has a specific direction that it moves in. And so in this ladder, or this portion of the ladder, we have our direction from the 3' to the 5' end. In just a second, we'll look at the opposite, the other side of the ladder here, so we have both sides. We can see what direction it's moving in. But again, a nucleotide is put together of a nitrogen base, a sugar, and a phosphate group. Those 3 portions, these 3 portions here, put together make up a single nucleotide. Multiple nucleotides put together make up DNA. Now, here's a better image, and we can actually see and look at 2 strands of DNA. So we have our double-strand molecule here. Again, we have our sugar, deoxyribose in DNA, ribose in RNA. We have our phosphate group, sugar, phosphate group, sugar, phosphate group. One of the ways that you can distinguish or tell or know exactly which molecule is the sugar is the nitrogen base, represented right here, is always attached to the sugar. So you can see all of these nitrogen bases for each of our nucleotide are attached to the sugar. So one nucleotide is composed of the sugar, nitrogen base, and phosphate, and let me identify one of those for you here. So right here would be a single nucleotide. We've got our phosphate, our sugar, and our nitrogen base, and so that's one nucleotide. And that's going to connect to another nucleotide on the other DNA strand. We've got a strand of DNA here and a strand of DNA here, and those are attached or connected to each other between the nitrogen bases. And what holds those together are hydrogen bonds. The hydrogen bonds are not as strong as a covalent bond or an ionic bond, and the reason for that, an important part of that, is it allows the 2 strands of DNA to separate. So when we get to DNA replication, when we start talking about transcription translation, these 2 strands actually break apart, and the hydrogen bonds, because they're not quite as strong as the covalent or ionic, allow that to happen. And so again, if we look at our direction, or the direction of how the DNA molecule is arranged, we have a 3' end here and a 5' end here. DNA is always moving in that direction, and so if you notice, our oxygen is flip-flopped from the side over here, and we're also moving from a 5 prime, from a 3 prime to a 5 prime. The arrangement of the DNA molecule is always this 3 prime to 5 prime. And so in order for that to occur on our anti-parallel strand, the molecule is actually flipped kind of upside down, or it's flipped over so that this molecule is still going from the 3 prime to the 5 prime end. We call this an anti-parallel arrangement, this arrangement of DNA, and so each strand of DNA is arranged in the opposite direction in relation to each other. And so if we go back and look at our image, we have 3 prime and 5 prime ends. They're arranged so that it's going 3 prime to 5 prime, 3 prime to 5 prime, and so they're flip-flopped in order to allow for this anti-parallel arrangement. One end of the nucleotide has a phosphate attached to the 5 prime carbon. The other has a hydroxyl group on a 3 prime carbon. And then, as we talked about, nucleotides between the carbon and phosphate are held together by phosphodiester bonds. And when we put 2 strands of DNA together, we get a double helix shape, and the holding of those 2 DNA strands together between the nitrogen bases is caused by or completed by hydrogen bonds. A couple other parts of DNA structure that we want to address is something called a nucleosome, and this is basically a coiled DNA into a bead kind of shape. Here in our image we've got our double helix, our DNA shape, and it's coiled around these proteins basically. And this is kind of a basic unit of packaging DNA. We see this when chromosomes are beginning to condense and to form. By using nucleosomes, it helps to regulate transcription, which we'll talk about in our next video. And a nucleosome is consisted of, or it's made of, DNA wound twice. You can see it's actually being wound twice around a protein, and this is composed of 8 histone proteins. And a histone protein is responsible for this first level of DNA packaging in chromatin. So chromatin is DNA and proteins put together. And so in this case, this is these histones and these nucleosomes are helping to condense and to shape the DNA into a chromosome structure. And so during mitosis, as we talked about, the DNA is condensed down into these chromosome structures to help it move and to be sorted during mitosis. Another part of DNA that we want to take a quick look at is introns and extrons. And for our purposes in our class, you basically need to know that they exist and kind of what they are. There are some really interesting new discoveries and scientific research going into introns and extrons. Basically, an exon is a sequence of DNA that's actually expressed and transcribed into RNA. And that RNA is then used to actually make the protein. So in our image here, we've got a DNA sequence. Here's an exon, intron, exon, intron. And so the only portion that actually gets turned into our messenger RNA, which then goes on to make our protein, is just these exon portions. The introns are parts that are not expressed and transcribed into RNA. And for a long time, scientists thought that it was just basically junk or garbage DNA. And it really didn't do anything. And in the last couple years or so, some new discoveries and some new scientific experiments have suggested that maybe those intron sections are actually responsible for some of the different expressions of the genes. And so there's a lot of research in that area right now, and it's kind of an interesting area to see how or what different parts of the DNA are actually used and responsible for making different proteins. For our purposes, for our class, this is the basic of what you need to know. Exon is a sequence of DNA expressed and transcribed into RNA. Intron is a sequence of DNA that's not expressed. The last portion that we want to talk about is the difference between single copy and highly repetitive genes. As we just saw in our last image here, not all of the information is going to be translated into proteins, and so not all the base sequences in DNA are translated into proteins. We have some areas called highly repetitive sequences and single copy sequences. Highly repetitive sequences are usually between 5 and 300 base pairs. So if we think back to our nucleotide, a base pair would be one nucleotide and its nitrogen base. So that would be 5 to 300 of those nitrogen bases connected together. It might be repeated. This sequence of 5 to 300 bases might be repeated 10,000 times, and generally it consists anywhere between 5 and 45% of the eukaryotic DNA. So it's a good portion. It's a large portion of the eukaryotic DNA. And it's not always translated. So we have this chunk of DNA, anywhere from 5 to 45% of a eukaryotic DNA that's not necessarily or not generally translated. The opposite of that is a single copy sequence, and this is translated. It occurs much less frequently, and it's a very small proportion of the eukaryotic DNA overall. Some basic information of the DNA structure. In our next video, we'll look at how transcription and translation works to actually use the information in DNA to turn it into RNA and then eventually into proteins.